Our next speaker is Jen Gommerman. She is the Canada Research Chair in Tissue-Specific Immunity. She is Professor and Chair of the Department of Immunology here at the University of Toronto. She received her PhD at the University of Toronto in 1998. She did a postdoc at Harvard Medical School, and then she joined Biogen as a staff scientist in 2000. And after 3 years in industry, she returned to academia here in Toronto as an assistant professor, and she's continued here and is now the chair of the department. Her basic research focuses on how members of the TNF superfamily of molecules regulate immunity and autoimmunity. Her team has uncovered a novel gut-brain axis that regulates neuroinflammation. But in the translational area, and again in the CoV-2 era, she contributed to understanding the mucosal antibody response to SARS-CoV-2 and resulted in her co-leading a biomanufacturing and pandemic preparedness effort for a hub of 8 Ontario universities. She's run a number of mentorship awards at the university, and as you'll hear, she's really a powerful player and a powerful spokesman for science. Jen.
Well, thank you for that very kind invitation. Bonjour à tout le monde, c'est un plaisir d'être ici. I'm so grateful to have had this invitation from the Gairdner Foundation, and I'm particularly grateful that I get to share this session with these amazing speakers. Congratulations to all the laureates, but especially to Dr. Kang, who's joined us today to give her second talk, which is, I think kind of badass, that you've been able to give 2 talks in 1 week on 2 completely different topics. It's really inspiring. Like Dr. Kang mentioned earlier, when you work on one pathogen, in my case also rotavirus, and then SARS-CoV-2 pops up, you think, well, I can't work on that, but you actually can, because some of the common elements to these pathogens is really the mucosal immune system. And I'm a basic immunologist, and I'm really interested in how mechanisms of how these immune responses are primed. And if we can understand that, we can potentially develop new types of vaccines that are more effective. And so I just want to get everyone on the same page. When I think about IgA and IgG, which are the antibodies that we think about as providing defense against pathogens, I think of IgG as sort of like the security guard inside of a dance club. So if you have an undesirable person who comes inside of the dance club and causes problems, IgG is going to take care of you. And it's going to use a potentially inflammatory mechanism to take care of you, whether that be complement engagement, for example. But when I think of IgA as the bouncer outside of the dance club that's going to prevent the undesirable person from getting into the club in the first place. And that's because IgA, first of all, IgA does not fix complement. It's more kinder, gentler antibody, but it's very strategically placed. And that strategic placement is at the mucosal epithelium, where IgA can be transported across the epithelium and then be put into, for example, the lumen of the gut, or the saliva, or the airways, or the breast milk. And so this provides a really great way of putting our immune system outside of the body. So to get everyone on the same page, I just want to remind you that IgA can protect against infection, and that's been shown in different viral settings, and I'll show you some data on SARS-CoV-2 today. And just to explain how this immune response takes place, what I want you to think about is that the priming of this immune response takes place in lymphoid tissues. In the case of the gut, these are the Peyer's patches and the mesenteric lymph nodes, which support a germinal center response, where B cells and T cells talk to each other in a very coordinated dance, and ultimately the byproduct of that immune response are plasma cells that make these antibodies. Now, these plasma cells don't do very much good if they're staying in the lymph nodes. They have to leave, and they have to go to where I would call the effector site. In the gut, this is the small intestinal lamina propria. For IgG plasma cells, these typically go to the bone marrow. We've learned recently in some very nice high-profile papers from Ziv Schulman's lab that the nasal turbinates can be a place where plasma cells live. And this is where they produce antibodies. And in the case of IgA-producing plasma cells, we know in humans that these IgA-producing plasma cells can live for decades. And that was established based on carbon dating. And so I think the gut is an unappreciated reservoir of long-lived plasma cells that I think would be very useful for vaccination strategies. So I'm going to talk, I'm a basic immunologist, so I'm not going to go too deep today because I want to cover some of the similarities and differences between the IgA response between these two viruses. I'm going to talk about the kinetics of these responses, their longevity, do they provide protection, and what variables can impact the initiation and sustaining power of these responses. So the first part of the work I'm going to show is Dr. Kei Hanuda, who's an amazing postdoc in the lab, who joined the lab with the objective of trying to understand what are the rules of engagement for generating an IgA rotavirus-specific plasma cell response. And even though we know a lot about IgG responses, we actually know not as much about IgA responses and what are the rules and regulations for how this works. And you've already heard about rotavirus. If you went to the talks yesterday, you would have heard from Dr. Kang that this is a pathogen of real concern, especially for lower middle income countries. And IgA is very important for protection against this pathogen. It's an enteric pathogen. It infects the small intestinal epithelium. And we got a mouse-adapted strain from Harry Greenberg's lab at Stanford, and that's what Kay is used to study this in mice. And he cloned two proteins, VP4, which is a surface protein, and VP6, and he expressed these proteins. They were very not well-behaved proteins, but he got it to work. And with those proteins, he made reagents, either proteins that he used for ELISpot assays or for tetramer analysis by flow cytometry. And what he found was that if he looks at that induction site, the Peyer's patches, IgA and IgG responses are really in lockstep. The antigen itself has slightly different kinetics, but overall, these two responses, IgG, IgA, happen at the same time in the same tissue. But when he looked at the effector site, and he did this in some experiments up to 200 days, what he found was that IgA actually comes up faster and more robustly as a plasma cell response. And it actually precedes IgG. So then he looked at, is this a long-lived response? And indeed, he can pick up tetramer-positive VP4 and VP6 plasma cells as late as 180 days post-infection. And by that time, these plasma cells are quite quiescent. They're not dividing anymore. And they have attributes about them that have been shown to be associated with a long-lived phenotype. Their B-cell receptor, he sequenced the IgA B-cell receptor on these plasma cells, and here he found something really interesting. So when he looked at different time points post-infection, the VP4-specific IgAs and the VP6-specific IgAs were all accumulating mutations. And this is indicative of an ongoing germinal center response, but what's interesting is that we know by day 14 in this mouse system that the virus is gone. And so this is an ongoing germinal center response that continues to percolate through time. And that's actually also been seen for SARS-CoV-2 by Michelle Nussensweig and other scientists. And they attribute this to a long-lived antigen trapping, potentially in the gut. So Kay actually cloned and expressed some of these antibodies and made IgA dimers with them. This is very laborious, but what he was able to show was that these antibodies had a range of affinity, and they also had the ability to neutralize the virus. And so if he took some of these antibodies and gave them to a mouse that couldn't make their own antibodies because they were immunodeficient, and then gave them the rotavirus infection, what he found was that many of these antibodies, but not all, could completely prevent the infection. And this is a negative control antibody here. This is a SARS-CoV-2 specific IgA compared to some of the rotavirus specific IgAs. And a few of them worked not as well, and this one didn't work at all. But you certainly can make neutralizing antibodies of the IgA class in response to rotavirus infection. So as I've already alluded to, we know a lot about the mechanics of how an IgG response is generated. And it starts with dendritic cells, myeloid cells that pick up the virus, and then they present that virus to a naive to CD4 positive T cell. That T cell becomes activated, and it takes on a phenotype called a TFH cell. And I'm sorry, immunologists have a lot of jargon, but TFH cells are really important, and we found out that in response to mRNA vaccines, TFH are really strongly induced. And they're very good at teaching B cells and helping B cells to undergo this affinity maturation process in the germinal center and ultimately emerge as plasma cells. So all of that is in our textbooks. And so we wanted to ask, are the same rules being played out for IgA? And the first thing we queried using a number of genetic approaches I won't show you here is whether TB interaction was necessary. And so we used knockout mouse models and pharmacological approaches to test that. And indeed, when you do that, you lose both the IgG and the IgA response. So no surprises there. But then we took a step back and we thought, okay, as a control, that's famous last words, as a control, we deleted class 2 from dendritic cells, and we figured that this would completely shut down the rotavirus-specific IgG and IgA response because you won't get this T-cell priming event. But in fact, what we found was that although the IgG response was very strongly inhibited by this genetic approach, the IgA response was just fine. And so that was a big surprise because that's not really what our textbooks teach us. We wondered if this IgA was just sort of garbage IgA, but it is indeed heavily mutated, the same way you would find in a wild-type animal. And if you take that IgA and you co-incubated it with rotavirus and then use that to infect an animal, it prevents infection, just as it would in a wild-type animal. So this antibody is functional. So this means that you don't need class 2 on dendritic cells to prime this response, which is unusual. And we later showed that if you simply express class 2 on B cells, that's actually sufficient to prime this IgA response. And so one of our working theories is that maybe this is why the IgA response is so fast. It's somehow going through an independent pathway that's generating this IgA plasma cells very quickly. The next question for us then was, so why would there be different types of immune responses occurring in the same lymphoid tissue? And so what Kay hypothesized was that maybe the T cells themselves are heterogeneous, and that some of these T follicular helper cells are important for helping an IgG response, but some maybe different T follicular helper cell is important for helping an IgA response. And immunologists love to divide up immune subsets. That's their favorite thing. It's not my cup of tea, but I realized in this case that he might be right. So what Kay asked me to do, he said, can I order 100 mice and infect them with rotavirus and fish out all the antigen-specific T cells and profile them? And by the way, I first have to map the class 2 restricted epitopes on rotavirus. So I said, go at it, fill your boots, and that's exactly what he did. So he mapped the class 2 epitopes that were we were able to then make tetramers. Mark Jenkins from the University of Minnesota made these for us. And so these are rotavirus-specific CD4 positive T cells, specific, sorry, the labels are missing, specific for VP4 and VP6. So this is a great reagent that hasn't existed before that we can now use to fish out rotavirus-specific T cells and ask what they look like. And so he did that. And when he did some analysis, he noticed that there were not one, but 2 types of T follicular helper cells. And the second type made this cytokine interferon gamma, which is unusual for these types of cells. Usually a T follicular helper cell is in détente with an interferon gamma producing T cell. They're usually kind of yin-yang. But here we had something in the middle. And when he got rid of these cells genetically, what he found was that the IgA response was strongly impaired, whereas the IgG response was intact. And so this supports this hypothesis that there's some heterogeneity or sharing of labor of the IgG versus the IgA response. And so when we think about making vaccines that induce these responses, I think we have to start thinking about the T cells and what they need to look like in order to support this response. And he went on and he showed actually what these TFH interferon gamma producing cells are doing, he was able to show that they were inducing a chemokine axis that endowed plasma cells with the ability to move to their effector site. And that seems to be what this program does. So our next question is, okay, if this is the rules of engagement for a rotavirus response in the gut, are the same rules at play when you have an airborne infection in the upper airways? And we don't have all the answers to that. It's difficult to do SARS-CoV-2 experiments in level 3. We are starting, and we've had lots of help from EPIC and from others here who've made that possible. But we do have access to human samples. And so we, at the beginning of the pandemic, thought, okay, we're mucosal immunologists. Everyone's really obsessed, rightly so, with getting serology assays working, where we look for IgG against SARS-CoV-2 to assess seroprevalence. What about the saliva? This is where the virus enters the body. Can we also detect antigen-specific IgA responses to the virus in the saliva? And there was reasons to look at the saliva because the receptors that are used for SARS-CoV-2 to enter epithelial cells are expressed in salivary glands. And I want to just remind you that the setup for a salivary gland is very similar to what you would have in the gut. You have this epithelial layer, and you have this polymeric Ig receptor that allows for IgA dimers to be transported across the epithelium, where they can then bind to commensal microbiota, and they can also bind to pathogenic agents such as SARS-CoV-2 or in the gut, rotavirus. So we developed an ELISA, and I think one of the themes that I think is so important from the Gairdner Week has been it takes a village to do complex interdisciplinary work. And that was certainly true of our efforts in SARS-CoV-2, where we needed to partner with people like Claude Gingras, who had a serology platform working, as well as our clinical colleagues to obtain the correct cohorts to study. And I want to make a really important shout out here to Dr. Gary Chow, who's sitting right there. When he was a project manager in my lab, he's now the research manager for our department. And he basically took a closet and he made that into a phlebotomy room. And we became a SARS-CoV-2 lab within a few weeks. And he really was the mastermind behind all of the studies that we did along with Laurie Seafried. And then I also want to make an important shout out to Salma Sheikh Mohammed, who's sitting beside Gary. She is just writing up her thesis now. She is on the job market, and she is amazing. And Bauletta Isho, who's listening in online for all of the work that I'm about to show you, as well as Eric Cow, who did some of this work too. So now focusing on SARS-CoV-2, what is the kinetics of the immune response in the saliva? And so to do this, we really wanted to capture the asymptomatic phase of the infection. And we know that's hard because you're walking around and you have COVID and you don't know you have COVID, so why would I take your saliva? So who do I focus on? Well, early on in the pandemic, Daryl Tan started a study called CoReprev, where he was doing a ring-based contact tracing model. And we collected saliva samples and nasal swabs every week from people who had been exposed to an index case. And we thought, oh great, we'll get tons of samples. But it turned out that in early 2020, everyone was following the public health recommendations, which was great. It meant that we had, there were less infections, but it was bad for our study because we didn't have so many people who acquired SARS-CoV-2. Nevertheless, we did get some, and what we found was that during that asymptomatic phase, like rotavirus, the IgA response actually precedes the IgG response in the saliva. In terms of the longevity, and this is work we did with Claude Gingras, we found that the saliva IgG and IgA response correlate well with the serology. But while the IgG response was relatively stable over a 100-day period, the IgA response was not. So the IgA response was actually quite short-lived to a viral infection in this particular group of convalescents. Okay, so what about vaccination? Once vaccines were rolled out, we worked with Alison McGeer and Sharon Strauss, who were running long-term care facility cohorts of healthcare workers, but it was hard to catch the baseline in those individuals because rollout happened so fast. And so once vaccines were available to a broader group, we started studies in local populations at the university, and we collected saliva from people who had been vaccinated. And what we found was, and this is, again, textbook and not so surprising, that one dose of vaccine gives you an IgG response in the saliva that's lower than what you get in convalescence, but with boosting, you start to approach the level of what you see in convalescence. Now, we had a bit of a surprise because we also looked at the IgA response and we thought there would be no IgA or no secretory IgA in the saliva. But actually, we could see a little bit. But just note here that this is really quite low, but it's there and it's bound to secretory components. So somehow this local IgA response is induced in response to an intramuscular vaccination. But it doesn't persist. And so after this first dose, you really can't recover it in most people. We've also looked at another cohort with Sharon Walmsley, where we've looked at vaccinated people who've then had breakthroughs. And again, the IgA response lasts about 90 days, and then it's gone. So vaccination and infection and breakthrough-induced IgA response is short-lived. Does this IgA response matter? Does it help? So we partnered with Alison McGeer as well as a group from Sheba in Tel Aviv that were looking longitudinally at people who had been vaccinated and then tracking breakthroughs. This is all pre-Omicron era. And this is the Sheba data, and what we found was that if we measured IgG to SARS-CoV-2, there didn't appear to be a difference between cases and controls, whereas IgA levels were higher in people who didn't get a breakthrough infection compared to those who subsequently did get a breakthrough infection. And this was mostly, I believe, to Delta. And two other studies have come out and actually looked at mucosal secretions, both of them from Sweden, and found that there is some advantage of having an IgA response in mucosal secretions. It's a protective factor for preventing breakthrough. It's not the whole story, of course, but it seems to be a protective factor. So what are the variables that impact the IgA response to SARS-CoV-2? Well, when vaccine rollout started to happen in children, we thought, let's have a look at kids and see what their response looks like. And here we did a Twitter campaign where we posted and said, listen, if you want to give us your spit then and maybe one blood sample, then we'll give you a tour of the lab and we'll give you a badge. And we had 100 kids in 1 week. And so this was our cohort, and it was just a really great experience in not only doing translational science, but in knowledge mobilization, and it was wonderful. And I'm only going to show you the IgA response here, but in general, the kids had way higher responses to vaccination than adults. But after the second dose, this tended to equilibrate. So it was really that first dose where they had a big response. So we became interested in what, this is a big range, and we wondered what types of variables could promote a mucosal IgA response. And remember I told you, and this is true of the gut as well, there's a commensal microbiota that lives in all of these mucosal tissues. In the gut, it's a very complex commensal microbiota. In the saliva, a little less so. And so we wondered what was going on with the IgA response to the commensal microbiota, both during infection, and I won't show you that data today, but also is this potentially a correlate of what your vaccine response looks like? And in fact, when we measure the amount of bacteria that are coded by IgA using this assay called bug flow, we saw a reasonably okay correlation between the amount of IgA-coded commensal bacteria a participant has at baseline versus how much anti-spike or RBD IgA that participant makes in response to vaccination. And this is in kids. And then if we looked at the amount of this IgA-coated bacteria and then subsequently, and this was based on self-reporting, monitored breakthrough infections, what we found, and this is the Omicron era, what we found was that kids that had an early breakthrough post-vaccination had the lowest levels of IgA-coated bacteria compared to a later breakthrough or no breakthrough at all during the course of this study. So what have we learned about these two viruses? Well, when we look at the kinetics, both of these viruses show us that IgA comes up really fast. And so I like to think of the IgA response at a mucosal surface as sort of the more primitive response, whereas the one we learn in our textbooks around IgG is more boutique. But really, I think vaccines that harness this quick immunity at the mucosal surface are really needed. The longevity of the response is different, though. If you have a breakthrough infection through the nasal passage or through the mouth, this is giving you a short-lived IgA response. And I don't understand why, and I think we have to understand that when we think about routes of delivery. Whereas the rotavirus IgA response is actually quite long-lived through the oral route. The protection, there is protection from IgA in both scenarios, and the variables I've just told you about. And those are the types of details we need when we think about vaccine formulation. And I've been really interested in the possibility of whether, for us to be prepared for the next pandemic, I would love to have a pill that we could give people or animals to protect them against a virus. And you might think that's weird because we've been talking a lot about aerosolized viruses, but we actually published before that IgA plasma cells that live in the gut not only live there a long time, but they can leave the gut. And we showed that this was true in response to influenza infection in mice. They can go to the flu-infected lung, they can go to the lactating mammary gland, and they can even go to the meningeal dura. And so I think this is really important, and we're excited about maybe putting all these findings together into new strategies. That's the lab, and thank you for your attention. I've mentioned these people as I went along, but in particular, I want to shout out the funding sources that helped us along the way. There was a lot of grants and a lot of cohorts, and that's what really fueled us through this period of research. Thank you.